So, welcome to hematology. This is going to be part one of a two-part lecture. It's going to revolve around hematopoiesis, or the generation of blood. This lecture is going to be solely focusing on what does it mean to do hematopoiesis, and what is the general process on how to develop blood cells as we know of them. Here are the general objectives. If you are able to achieve these objectives, then you're going to be on a very good course. So, blood cells have a decreased lifespan. Unfortunately, we haven't figured out a way to achieve immortality yet, in the same way as with our cells. Granulocytes, or our white cells and platelets, will only live for about a week. Red cells will only live for about 120 days. And this is a good thing. We need to have this cycle between the old, mature, and circulating blood cells. That way we can have fresher and better cells being produced at the same time to replace them at a healthy rate. If we didn't get rid of old maturing cells, then we would all just kind of swell up like balloons, and I don't think anybody wants that. So, I love root words. I tell my students this a lot, and I genuinely found that it helps me when I was trying to understand the course material. Heme, meaning blood. Poiesis, meaning generation. So we are solely talking about the generation of blood. The formal definition is to just replace maturing red cells by maintaining an adequate number of cells to carry out life functions. Cell proliferation, differentiation, and cell death will work together to help meet bodily demands. Anything that is in your peripheral blood, or anything that is circulating throughout your body outside of your bone marrow and outside of your organs, should be mature cells. The definition of mature cells, meaning that they are functional, and this is key word, functional cells that are incapable of re-entering the cell cycle to actively mitose or divide again. Again, these are functional cells that will not divide once they have fully matured. In adults, the blood cells are being made in your bone marrow. It's a giant amount of red cells and white cells and platelets being made each and every day just from you existing. However, we humans are designed to live, to survive in case something happens to us. So, because of that, sometimes you get certain diseases, sometimes you get certain conditions. Well, your body, when that happens, will naturally want to compensate for that to happen, or make up for the fact that you have a disease or a condition. Easy one to think of is any time that you get an infection, your white cells are going to increase in response to that infection. If you're losing blood, or if you're under anemia, then we're going to want to produce more red cells to stop anemia or try to make up for the anemia. So, as your cells are replicating in the bone marrow, they have to differentiate and specialize. Well, how do they do that? Well, that is done via cytokine stimulation. What cytokines will do is bind to the cell membrane, and then once those receptors are bound to, that's going to cause gene expression, which will then create unique cell lineage. That being said, commitment must occur before differentiation could occur. Commitment is basically the process of two daughter cells who originally came from the same line, but once they divide, they then start undergoing different routes or different paths down their maturation or differentiation pathway. Once a cell has been committed to a certain lineage, then that cell cannot jump right back over. Your lymph cells are never going to end up becoming your red cells, and vice versa. Once they have been determined to be that cell lineage, they can't swap. So, as the cell starts developing from your stem cells, then that will commit and differentiate to become the different types of cells that we can then identify in your body. So we're going to talk a lot about growth factors and cytokines. Cytokines, by definition, are just proteins that are released by cells, which will then affect the interactions and the communications between the cells. They can only work if they can bind to a receptor that is present on the target cell. Once they bind, then it's going to cause an intercellular cascade that will cause one of four effects. First is survival, or proliferation, or differentiation, or apoptosis. So I'm not going to go super deep into this. I'm assuming that you are going to be a med tech because this is what the course was designed for. But basically what happens is once that cytokine binds to that receptor, it's going to start a pathway. That pathway is fueled by protein phosphorylation, which is basically the process of transferring phosphorus groups over to the proteins or receptors within that cascade. So when that happens, that phosphorylation will either activate or deactivate proteins, which will either help propagate or dampen the original cell signaling. If that cell signaling isn't what we want it to be or would be considered inappropriate, then that could lead into cancers and diseases. So this is just a really, really simple image, right? The L circle there is meant to be a cytokine binding to those pink receptors. Once that happens, that's going to cause a biochemical change in those receptors. Once those receptors experience that biochemical change, then that's going to cause this cascade where the phosphorus groups is going to get transferred to the next protein, to the next protein, to the next protein until it reaches the nucleus. Once that amplified cell signal reaches that nucleus, the nucleus is going to do something or activate. And once it activates, then that's going to form the cell response. But at the end of this intracellular cascade, the goal is to induce gene expression. And by inducing a gene expression, you're going to cause new proteins to be made. When new proteins are made, then that will always lead to a new function. Remember, genes lead to proteins, proteins lead to function. So what this whole thing is just trying to describe is that we require this complex cascade of signaling, phosphorylation, dephosphorylation, and activation of those intercellular proteins that will then activate transcription factors in the nucleus. We're just trying to generate some sort of change in order to induce a change in the nucleus. Growth factors and cytokines together will govern your hematopoietic stem cell survival, self-renewal, proliferation, and differentiation. The most common types that you'll see here are going to be called interleukins, colony-forming units, erythropoietin or EPO, and thrombopoietin, TPO. Growth factors, by definition, are just a molecule that will affect the growth of the cells. But over time, we've just kind of included molecules that will either help promote or inhibit mitosis or affect cellular differentiation in some sort of way. Just sort of keep in mind, this is almost like a all squares are rectangles kind of situation, but not all rectangles are squares. All growth factors are cytokines, not all cytokines are growth factors. So what the cell needs in order to be able to survive is having this continuous supply of growth factors. Most cells, by design, are unable to promote their own proliferation and need some sort of external stimulation in order to be able to continue surviving and growing. If there's no longer any growth factors being supplied to that cell, then it's going to lead into apoptosis and death. Growth factors will either suppress apoptotic signals or promote proliferation. But they're both getting to the same thing. It's going to tell the cell to continue living on or don't die. There's a lot of differences and similarities, but they're all just going to go to the same effect. The differences and similarities might be within their structure or what cell they act upon. Most cells will be produced by stromal cells that are found within the bone marrow, but the only exception that we're looking at is the EPO, erythropoietin, which comes from the kidney. Growth factors are what we call pleiotropic. Pleiotropic just basically means either the same growth factor will exert different effects on different cells, or different growth factors can exert similar effects on different cells. This allows them to work synergetically in order to change or cause some sort of effect. In most growth factors will reach hematopoietic cells via the following pathways: autocrine, paracrine, juxtacrine, endocrine, or exocrine. Though most of the time in your blood cells, it'll occur via paracrine or juxtacrine signaling. Here is just a list of the pathways that I was just talking about. Autocrine basically means it's acting on the same cell. Paracrine means it's going to act upon its neighbor. Excuse me. Juxtacrine means that there is a different. It's kind of weird. Let's see. So let's say that we have cell A. Well, cell A can't is not going to have its neighbor act upon it for some reason. So this is where cell B and cell C come in. Cell C can't directly interact with cell A for some reason. So cell B will then interact with cell C. Once it's able to interact with cell C, it's going to tell cell C, hey, go interact to cell A for me. So then cell B will go ahead and act upon cell A. And that's juxtacrine. You'll see a lot of that in immunology. Endocrine means that there is a cell trying to communicate to another, but it's on the opposite side of the body. It needs to travel through a long distance in order to be able to get to its target. Exocrine means that it's almost a similar process as endocrine, except the signal has to leave the body and then come back in to be able to exert its effect. Really common example is pheromones that most animals will use in order to communicate with one another. We also, in conjunction with positive regulators or signals that will cause the cell to continue proliferating, we also do have negative regulators, and they're just as important as our positive ones. Negative regulators just represent a group of polypeptides that will inhibit cell proliferation. So this will either decrease the production of stimulating factors or increase inhibitory cytokines. So the most common ones you'll see are the transforming growth factor, P16, P21, interferons, and TGF-beta, or tumor necrosis factor alpha, which is a really common one that you'll see in molecular biology. If you prefer images, this is a really nice way to be able to show what happens. So if I want you to literally just remember one slide, because this is literally the core of the development of your cells in hematology, this is probably it. We're always going to be going from stem cells, which will then develop into progenitor cells, which will then develop into maturing cells. Remember that our goal is to always get new cells to replace the old ones, and we need those new ones to properly develop into maturing cells or mature cells. So we're going to talk about the pluripotent stem cell. Or you'll also see the term, since we're talking about hematology, is the hematopoietic stem cell. So every single blood cell that we're going to talk about comes from the hematopoietic stem cell. we will never be able to tell hematopoietic stem cells apart from one another. Um, it once it looks all the same at a certain point. But stem cells sort of have this unique property of self-renewal and regeneration as they asymmetrically replicate. So this is not something that most cells are able to do. So we're familiar with mitosis, where you get two identical-ish daughter cells. Well, that's meiosis, but mitosis would be two identical daughter cells. In asymmetrical replication, one of the daughter cells is going to be different. So we're always going to get an additional stem cell out of it that will resemble the mother cell. But the daughter cell will undergo cellular differentiation and will not become the same mother cell that it came out of. And that will keep replicating until the, well, end of life, really. Again, the thing is though, we humans aren't, surprisingly, not very good at being able to tell um, stem cells apart from one another because we don't have microscope eyes. They'll just always look like this. They'll just look like big cells with giant nuclei in them. So the smarter thing to do is to use flow cytometry. By using flow cytometry, we can differentiate those precursor cells via CD markers. Their full name is called cluster differentiation. But basically, it just means the proteins that are found on the surface. CD34, you'll probably hear this popping up a lot as you stay within your hematology course. CD34 is really important in being able to identify early progenitor cells. Um, it's basically the biggest way to confirm that if a patient has any sort of blast circulating throughout their system. And blasts are in high amounts are really strongly associated with leukemias. The methodology of cell flow cytometry is this. Basically, we get cells that pass through a single point, and that single point is hit by some sort of light or laser. Well, as they're passing through, these cells are marked with fluorescent markers that will attach to specific CD markers that we need them to attach to. As the cell passes through, then the light scattered can be used to identify size, number of cells, and granularity of the cell. If we have a fluorescent marker on there, then the light signal will also excite or fluoresce the markers that are attached, and that will help us mark out where that cell is based on where the chart is. So stem cells live in specialized regions within the bone marrow called a niche. We have two big main niches. One is the osteoblastic and the other is the vascular. Um, the osteoblastic niche is dedicated to maintaining hematopoietic stem cells, and it helps balance that self-renewal. Vascular niches is near the endothelial cells that will provide and respond to chemical signals. So we're going to move down into our colony forming units. Once hematopoietic stem cells commit, they then become progenitor cells. Progenitor cells don't possess self-renewal, but they still look the same as hematopoietic stem cells. They are more mitotically active, but we wouldn't be able to tell that apart because we don't really film these in live camera. So eventually, those hematopoietic progenitor cells then form into colony forming units based on the cytokines, growth factors, and colony stimulating factors supplied to that colony forming unit. So for example, if we give a hematopoietic progenitor cell a CFU-GEMM, then that will cause that HPC to become a granulocyte, erythrocyte, monocyte, or megakaryocyte, but it will not become a lymph. As those colony forming units continue to develop, then they start to take on more morphological changes that we could see. What's happening here is that it's going through these different gene expressions as it's continuing to develop, which are induced by those different cytokines. Once those gene expressions are active, then that causes protein expression. When protein expression occurs, and that will cause functional changes, then which will then eventually lead into identifiable cells that we can actually tell apart now. Maturing cells should take up about 95% of your cells in your bone marrow. Key note is that mature cells cannot proliferate, replicate, or self renew. And this is again by design. That way, we can always replace cells as we need to, and we're also getting rid of cells at the appropriate time. So that way, we're not going to swell up like balloons. So, if you're a tech, we again cannot tell cells apart based by just looking at them. They'll have this one characteristic of looking like a giant cell with a giant nucleus inside of it. But we cannot tell these specific types of cells apart. The earliest precursor cell that a tech will ever call is a blast. Um, Blast will always is considered a very immature cell. Their job is to continue to mature in the bone marrow and eventually will become fully functioning cells. The thing is though is that they should not be seen in peripheral blood. If we do see them in high amounts, then that's associated with some sort of disease. Biggest one that we love talking about is leukemias and lymphomas. So that potent stem cell that we talked about will either become the myeloid or lymphoid progenitors. Once they have become either myeloid or lymphoid, they will not swap over. The lineage restriction occurs right at this step. And again, we talk a lot about these apart. The colony forming units, the cytokines and the interleukins will drive the cell differentiation and maturation processes. Myeloids will always give rise to granulocytes, erythrocytes, and megakaryocytes. Granulocytes are made up of white cells such as the neutrophils, eosinophils, basophils, and monocytes. And the common lymphoid progenitors will only give rise to lymphocytes, which have the subclasses T lymphocytes, B lymphocytes, natural killers. So I kept talking about interleukins a lot, and here are a few that are important for you to know. Interleukin 3 will give rise to erythrocytes. CSF-GM will give rise to myelogenous cells, except for basophils. Interleukin 6 and interleukin 11 will sort of touch base on a lot of things, but we usually talk about it a lot in platelets and megakaryocyte production. So those are the earlier growth factors. As the cell continues developing, we'll start talking more about the late acting growth factors. So CSFG gives rise to granulocytes, CSFM will give rise to monocytes, EPO is the hormone that will increase red cell production, and remember it's only made in the kidneys. TPO will regulate platelet and megakaryocyte production. Interleukin 5 will give rise to eosinophils and granulocytes. If you love talking about interleukins, this is a really nice chart to look back on. I'm not going to read all these to you, fortunately. We're not going to torture ourselves that way. Alright, now we get to talk about the later bits. Erythropoiesis, again, love my root words. Erythro means red blood cell. Poiesis means generation. So we're specifically talking about cells that are found in your erythroid line. So the CMP will eventually become a RBC precursor called BFUE. It's stimulated by interleukin 3 and CSFGM, If we try to apply EPO at this stage, it won't work. So it takes about 14 days for it to mature. Once it matures from the BFUE, it becomes a CFUE. This is also called, or CFUE. Eventually, CFUE will develop into the first recognizable precursor of RBCs called pronormoblasts. At this stage, it's influenced by interleukin 9, interleukin 11, and erythropoietin. Granulocytes and monocytes come from the GMP, which comes from the CMP, as well as the BFUEs. Cytokines CSFGM and interleukin 3 will drive differentiation from the CFU-GEMM. The GMP will either become a granulocyte or monocyte, and that will be induced by either a CSFG or CSFM, respectively. Again, your granulocytes are going to be your basophils, eosinophils, and neutrophils. Eosinophils are also derived from the GMP, but they're under the influence of interleukin 5, as well as interleukin 3 and interleukin 4. Thrombopoiesis, the developing of thrombocytes or platelets. Platelets are the functioning peripheral blood formed by the root element, formed to be able to clot things. And those come from the megakaryocytes in the bone marrow. Megakaryocytes are formed from MAPs, which are formed from CMPs, and the cytokines that drive this development is interleukin 11 and thrombopoietin. Lymphocytes are derived from the CLP, and lymphocytes get to be a little bit special. So all the cells that we just talked about develop solely in the bone marrow. But lymphocytes have to go on a journey. They actually have to travel out of your primary lymphoid organs, which are the bone marrow and the thymus, and then develop into the secondary lymphoid organs, which are your lymph nodes and your spleen. Interleukin 2 and interleukin 7 are for developing lymphocytes. If you want to go by one chart just to memorize your CD markers, this is a really good one. I would recommend just looking at this one, drilling it into the brain. And that's the end of the presentation.